I'm going to go ahead and get started. We have a class that is supposed to come into this room right after, so I want to try to go ahead and get us started on today's seminar. It's my very great pleasure to get to introduce today's CTSI distinguished speaker, Dr. Brian MacVicar. Brian completed his early training at University of Toronto, where he earned his bachelor's, his master's, and his PhD. And as an electrophysiologist during graduate work, he contributed to discovery that some of the neurons in the hippocampus are actually gap junction coupled to one another. They have electrical synapses. He completed postdoctoral training at Michigan State and NYU, and then started his independent lab at University of Calgary. And basically right from the start was really making a lot of very impactful contributions to a number of fields. In particular, his group was one of the first groups to show that astrocytes, a key form of glial cell, are not as passive as was previously thought. So these glial cells, sorry, I'm getting a little nervous because the screen changed there. These glial cells can actually express voltage-activated ion channels and neurotransmitter receptors that are present in neurons. So that was really something that fueled a big upsurge in interest within the field in understanding the properties of glial cells. And Brian's group right from the beginning was interested not only in the properties of the glial cells, but how they might talk back to and influence neurons. In 2003, he moved his lab to University of British Columbia, where his lab is today. He is a professor of psychiatry at UBC and also holds the Canada Research Chair in Neuroscience at UBC. And I have to confess, Brian, you've been one of my scientific heroes throughout my training. Throughout his work also at UBC, Brian's group has made a number of really fantastic contributions to multiple fields. I really admire the blend of electrophysiology and cutting edge imaging approaches that his group leverages for their work. They've also contributed custom image analysis software development that they've then shared with the field. And I'll just highlight a couple discoveries that have been really noteworthy for the field. So Brian's group has made some really important contributions to our understanding of how astrocytes can influence vasodilation and vasoconstriction. So really shaping the supply of oxygen and nutrients to active circuits. His group has also shown how neuroinflammatory changes in microglia can affect synaptic function and elucidated some of the molecular players involved. And also key findings in the field of stroke, showing that pericyte responses during stroke are important for blood vessel regeneration. And connexins and pannexins may also be key molecular players in some of the damage to neurons following stroke. So just very consistent, impactful findings for our fields. There are a number of honors and awards I could point to, but I don't want to steal time from your talk. I'll just mention that Brian is also a fellow of the Royal Society of Canada and a fellow of the Canadian Academy for Health Sciences. So very pleased to have you here today and looking forward to hearing about what your group is up to.
Thank you very much, Lindsay. That was a very, very nice introduction. Thank you very much. I had to come back more often. Okay, so okay, all systems go. Okay, so a pleasure to be here, and it's a great honor to be a distinguished speaker. Thank you very much for that introduction, as well as the chance to come and meet you all, spend the day here, talking to Bal and Michael and the rest. It's been terrific seeing what's going on at UCLA and having a day to visit California. So I'm going to talk to you today about some work that we've been working on extensively, but I also want to maybe set a bit of an outline here. So I'll tell you first why I started working on glia. Then maybe this is a story for the trainees starting out. I'll talk a bit about a complexity of surveillance by microglia that we discovered a number of years ago. And I'd just like to review that to keep that in the awareness of the glial community. And then I'm going to deal with focus. The main focus right now that we're looking at is the links between Alzheimer's disease and glial activation and oxidative stress. And I want to argue and present the evidence that's in the literature that this is a very important target. Maybe this is part of the missing link, understanding the impact of A-beta that leads to dementia. A lot of work still to be done. I'm going to show you how we've looked at reactive oxygen species generation and tested the hypothesis that microglia cells are this critical generator of oxidative stress in Alzheimer's. And I'll first, though, show you evidence that the microglial cells can indeed produce reactive oxygen species to induce a form of long-term depression. Just to show you that they can do it, and it'll set the stage for us asking whether or not they actually do this around plaques and Alzheimer's type of condition in the mouse. We use ROS imaging using genetically encoded ROS sensors. I'll describe this, how we use this to look at microglial cells, oxidative stress around plaques. And I'll end with our early results. Our next direction is going to be looking at these issues induced by Alzheimer's type conditions in neurospheres using iPSCs, which I think may be a very powerful way to go in the future. Okay, so I've shown this before at glial meetings. I love to show, kind of brag on how glial cells are taking over. You know, Synapse had a lead, of course, had a lead for, there it is. Synapse had a lead. Everybody knows synapses are perfect, are important for running the CNS. Had an early start a long time ago. And but you know what? What's taking over is the growth of glial work, and astrocytes have surpassed this work on synapses. This is PubMed papers per year. And microglial cells is, I wish I'd started microglial cells earlier, because they have really accelerated and exceeded astrocytes as well as synapse. And I also want to touch upon the point, there's new concepts coming out with metabolic reprogramming, that the metabolism is related to microglial function and immune function. This idea of immunometabolism is taken off in diabetes, and it's just starting in the brain. So there's metabolic influences of immune function impacting microglial cells that is going to be quite important in the future. So my own trajectory, I started this at this time. My first paper as independent scientist with my own lab was in 1984, and I tested the idea: do astrocytes actually have ion channels? And this was kind of an issue that tell my colleagues down the hall working on glial cells, they roll their eyes. You know, people thought glial astrocytes are the styrofoam of the brain. They're only packing material. But as Lindsay mentioned, when I did my PhD, I looked at electrotonic coupling synaptic interactions in the hippocampus, and this was modeled by number of labs, Traub and Wong, et cetera, at this time in the early 80s, that this synchronization underlying epilepsy may be generated by local synaptic circuits. It's still an area of research. But what influenced me at the time was some recordings by Bruce Ransom, another real pioneer in glial research. He had a paper that I couldn't just forget this work. This is in the 70s, where recordings from astrocytes in the in vivo brain, if seizures was induced, astrocyte membrane potential changes very faithfully followed the extracellular recordings, extracellular indications of epilepsy in the intact brain. So I mean, it was a crazy idea. I thought, what if they actually have voltage-gated ion channels? What if they have transmitter receptors? What if the astrocytes are actually players in responding to activity around the surrounding nerve cells And maybe they're contributing to epilepsy in some unknown way. So this is a totally different era. For the students here nowadays doing research like this in the early 80s, this was to get a paper in science, where we fortunately I got a paper in science. This was not like science nowadays. We didn't have to do multiple data sets. You could actually do one simple, clear experiment and get a paper. And so I did this experiment, and it seems almost naive and simplistic now, but this is glial astrocytes in cell culture. It showed it's an astrocyte with GFAP staining. We impaled the cell with 2 electrodes and showed you could evoke a calcium spike. And this is the era when people argued, are there 2 voltage-gated calcium channels or 3? And I saw these vicious arguments between scientists. How many voltage-gated channels are there? So nonetheless, I was lucky to get this published, and particularly because reviewers' comments said, the author should show the functional impact of calcium signaling on astrocytes. And I would argue we're still working on it. In particular, so this is, we then pursued several different types of channels in the astrocytes, calcium activated potassium channels. They have neurotransmitter receptors. But working and solving this importance of calcium, it took the development of new technology. So it's when we had 2-photon imaging, we could look at calcium changes being triggered by uncaging calcium in an astrocyte, and see the calcium wave propagate through the tissue using this is not genetically encoded. This is before the days of genetically encoded calcium indicators. This is a Fira stain. And it was remarkable that calcium, in this case, had a very potent effect on causing vascular constrictions. And this led, of course, this is then controversial. Some people saw dilation. And we had another Nature paper a few years later explaining how dilation occurs. So there's a complex interplay with astrocyte calcium in terms of causing constriction under some conditions and dilation under other conditions, depending upon the metabolic state of the tissue. So it's at the time that we were doing this uncaging, we could see that you uncage to induce a calcium signal among astrocytes, and sometimes the power is too high. And Sean Mulligan, the student at the time, we'd see things kind of moving around. And this was a little surprising. And sometimes you can see lymphocytes move through the tissue, but if you do a lesion, you see kind of movement in the tissue, which is puzzling. We started to wonder, how can we address this question? It's the whole field exploded when Nimmerjam et al., and in Helmchen's lab showed that the microglial cells are constantly immunosurveilling surrounding tissues. So this has totally transformed the study of microglial cells, that these are active, they're responding, the cell bodies have these processes that extend and retract. I'm sure you've all seen these movies already, but I'm going to show a highlight maybe you're not so much aware of in the next few slides, is that there is a fairly large several micron processes that extend, retract over a kind of a minute time scale. And in Nimmerjam's paper, this is work from a student in my lab, it's an example following their lead. But what they showed, it's just beautiful work at the time. If you lesion the tissue, you activate a response of microglial cells where they're hunting for ATP. They have P2Y12 receptors on the processes. They orient, their polarization points toward where the ATP is being released by the damaged tissue, and these processes converge on this area of damage. And this is some nice work that a student in my lab, Dissing Olsen, did. He flash fixed these and showed that these P2Y12 receptors are in these growing blunt tips that are going toward the lesion. So part of what microglial cells are doing, these processes are surveilling around the tissue. They're hunting for ATP, but there's more to that. And I want to focus on another aspect that's also part of their surveillance. Is they're not only hunting for ATP as signs of damage or apoptosis. Apoptosis, these processes cause membrane breakdown. You get release of ATP and ADP that activate on the P2Y12 receptors. They're also trying to contact sites to recognize the eat me signals. So this is the call me signal, the ATP, but there's also eat me signals where they contact phosphatidylserine in the surface and is recognized by receptors on the microglial cells. So I would argue that this actually uses a slightly different search strategy. And in a paper that describing some work that Louis-Philippe Bernier, terrific research associate now in my lab, described, looks like if you look at these large processes that are retracting and growing, these are a few microns across, but there's also a fine filopodia coming off these processes that have not really been described. And we would argue that these are important for very fast searches looking for areas of contact. More work to be done, but I think that this represents a second stage of microglial searching, looking for contact sites, looking for eat me signals. An example of what this looks like in terms of scale is okay. So these are the large processes that are retracting and extending at a rate that's much slower than this fast filopodia that are scanning around this tissue, around the ends of these processes. And just to show you again. The tips and these colored dots are showing the distribution of the ends of these filopodia over a 5-minute period. So they're very rapidly scanning. I need to show that behind that slide. Okay, so this is not sure how to get rid of that now, but nonetheless, so the processes are moving 10 times faster than the large. The diameter is about 100 nanometers versus the processes typically of the slow-moving processes are several microns, and the distribution of the nanofilopodia that are scanning the tissue doesn't have the same polarity as the actual large processes growing out. And so within a few minutes, they are intensively scanning a larger volume at the very end of the tips of the processes. And our work in that paper showed that the cyclic AMP, intracellular cyclic AMP, is an important modulator of these nanofilopodia scanning. As that normally the baseline surveillance, large process, this outgrowth is driven by ATP, ADP acting on P2Y receptors. They decrease cyclic AMP, and this causes a retraction of the fine filopodia. When it's stable and the ATP is not activating on the receptors, cyclic AMP increases with the action of nitric oxide synthase as well as noradrenaline, and this increase in cyclic AMP increases the scanning at the tip of the of the process. And in some circumstances, when there's decompartmentalized cyclic AMP processes of retraction, there's very intense, rich filopodia scanning of the surrounding tissue. So our argument is, our hypothesis is that this type of scanning is reminiscent of this argument of levee flight as a very effective way to hunt for prey. This is found in foraging. It's the most effective way to forage for prey, and it's used by fish in the ocean, as well as in predators that are hunting for food on the savanna. It's most efficient to move quickly to a new area and then scan at a very fine scale within an area, and then go to another area quickly and scan at a fine scale. And this is shown by papers on this Nature paper in 2010. It's called levee flight foraging hypothesis. This is the most effective way to hunt for prey for animals. And I would argue this is the most effective way for microglial cells to find the targets, not only that have called them there, but also that they need to eat. Okay, so I'm going to move into the second phase of the story in terms of looking at astrocytes and microglial cells and the interaction with Alzheimer's disease. To summarize just globally what astrocytes do, they protect neurons from oxidative stress, with a system that uses their production of glutathione and cycling of the antioxidant ascorbate through nerve cells. So astrocytes have a very important role in controlling oxidative stress through this glutathione ascorbate pathway. And microglial cells, as the immune cell of the CNS, have been proposed to be the generators of oxidative stress, particularly in Alzheimer's disease. And in the initial work by Alzheimer's, of course, in his first study, he showed that the plaques, as he described in his first paper, are surrounded by glial cells. And our question was, is there a contribution of microglial and/or astrocytes to Alzheimer's neuropathology? And what led us into this line of experiments was the observation that there are non-demented patients that have Alzheimer's neuropathology. And this is called NDAM. And in the last 20 years, there was a lot of postmortem studies showing that a lot of people have Alzheimer's pathology, A beta plaques, but do not have dementia. And the study is looking at this Alzheimer's, this postmortem neuropathology. This is the first report, showed that in areas where you have plaques and Alzheimer's disease with dementia, you have lots of microglial cells. Microglial cells has proliferated, as well as astrocytes. Kind of the critical factor that's altered is the increase in proliferation of an activation of astrocytes and microglial cells. So, so I'm going to show you first that evidence from the lab showing that microglial cells can truly produce reactive oxygen species under some circumstances. And then we'll show the analysis of oxidative stress being generated around the plaque to see if the microglial cells are the players generating this. So the first experiments we did to find microglial cells can generate reactive oxygen species under a certain defined condition was inspired by a lot of the work in the literature that neuroinflammation and hypoxia act synergistically to generate CNS damage. A number of conditions, stroke, trauma, neurodegenerative diseases, there's a combination of both neuroinflammation and hypoxia. And that in the peripheral system, evidence has shown that these pathways show common pathways resulting in enhanced inflammatory reactions. So our experiment reported here, and this is to convince you that microglial cells can indeed produce reactive oxygen species. We did a very simple experiment, straightforward. Can we combine an inflammatory stimulus like a lipopolysaccharide, component of the outer membrane of bacteria, combine this with hypoxia, and see if there's a long-term effect on synaptic transmission that's related to the generation of reactive oxygen species. And a simple experiment in a way for electrophysiologists, we did synaptic activation of the CA3 projections of CA1 and look at field potentials here to assay the change in synaptic transmission. So this is the experiment. If you just put on LPS, there is no alteration of synaptic transmission. If we put on just lower oxygen from normal high oxygen to low oxygen, there's a transient depression that recovers quickly. And this is completely due to the transient release of adenosine from the tissue when during this transient hypoxia. Now, the surprise was when you combine the two, which is certainly something that enhances damage from inflammatory stimuli, combined with hypoxia, we found that now instead of recovery back to baseline, there is a appearance of long-term depression. So the field potentials normally shown here are not altered if there's no LPS with hypoxia. And there is a long-lasting decrease in the amplitude when the two are combined. This is not the typical LTD generated by NMDA receptor activation because there's no effect of blocking NMDA receptors. This effect is not dependent upon adenosine receptors. We could use the adenosine inhibitor to block this profound depression from the adenosine release. We still have long-term depression. So what's the mechanism? So then we looked at, this is nice, but doesn't actually, of course, indicate it's a microglia-mediated event. We looked at potential targets for LPS that could explain this generation of reactive oxygen species. LPS is a ligand for toll receptors. That's the canonical ligand for the toll-like receptor that's highly expressed in microglial cells, toll-4, but it's also a ligand for several other receptors, such as the complement receptor 3 and some other scavenger receptors that are on microglial cells. So we first tested the idea that this is a role for complement receptor 3. So CR3 is a combination of two transmembrane proteins. It's only in microglial cells. The receptor is disabled by using the CD11B knockout, so only CD18 is present. Microglial cells look fine, but they don't have functional complement receptor 3 receptors. And we used a knockout to see what happened to the effect of LPS plus hypoxia. And I'll show you in schematics what's going on, and I'll show you the data at the end. What we showed, and I'll show you the convincing data here, is that LPS and hypoxia causes a release of a cloud of superoxide around a microglial cell, that this acts on the GluA2 AMPA receptor, causing internalization, and this is the cause of a long-term depression. So the proof that this is indeed microglia and that this is indeed the complement receptor 3 is that, in contrast to normally this combination of LPS and hypoxia causes long-term depression, this is abolished in the CR3 knockout mouse. It's got to be microglial cells and it's evolving the CR3 receptor. And it's not the toll-4 receptor, the canonical LPS receptor, because the LTD still was unaffected by knocking out the toll-4 receptor. And it is long-term depression, the classical long-term depression triggered by a second mechanism, because we could block this internalization and this long-term depression by using a TAT interfering peptide, which crosses a membrane, TAT GluR2-3Y peptide or GluA2-3Y peptide. It blocks this tyrosine kinase cluster that's required for the receptor internalization on the tail of the GluA2 membrane subunit. That if we block this with a TAT interfering peptide, this prevents the internalization. And this is used in vivo to show that a phenomenon is in fact due to long-term depression because it blocks even LTD in the intact brain was injected into the live awake animal using a TAT interfering peptide. And what we found with recordings when the TAT GluR3Y peptide was applied, that the normally the LTD is at this level, the long-term depression was totally blocked from the application of LPS and hypoxia. And then this is quantified here. And to further convince you that this is a synaptically mediated event, is we did the mini analysis, looking at the mini amplitude that the hypoxia and combination hypoxia and LPS led to a substantial decrease in the average mini amplitude, that this was blocked by having the GluA2-3Y peptide in the pipette. So this is a localized applied through the wholesale voltage clamp recording. This blocked the development of the long-term depression from this combination. So the mechanism is activation of complement receptor 3 drives forward this pathway to activate the formation of an active NADPH oxidase complex. And the hypoxia drives forward the formation of lactate within the microglial cell, and this drives forward the development and formation of NADPH, which is feeding the NADPH oxidase pathway. And we show that this is the enzyme required here because we could block this generation of superoxide as well as the LTD by inhibiting the enzyme with a pharmacological inhibitor. And we could block it with a TAT-FOX interference peptide, which prevented the formation of an intact oxidase enzyme. It could also be mimicked by just applying lactate to the tissue. Lactate uptake into the microglial cell was sufficient to drive this forward pathway if we combine it with the CR3 application. We could also mimic the formation of long-term depression by applying a superoxide generator system using xanthine oxidase with trigger LTD. And if we use a scavenging agent, ascorbic acid, to scavenge the superoxide, this will prevent the development of long-term depression. So we have, hopefully, I've convinced you that the microglial cells, when appropriately activated, can generate a localized burst of reactive oxygen species. And this is sufficient to have quite profound effects on the surrounding tissue. At the time, we used imaging strategy to look for the also further proof for oxidative stress. This is a compound that gets trapped within cells. You end up with dichlorofluorescein diacetate that's within cells. These are nerve cells that gradually show increased fluorescence as the ROS is being generated, and it's leading to this compound being trapped. So this is kind of state of the art, maybe 10 years ago, to look at degrees of oxidative stress. Well, this is not sufficient if you really want to look at variations of oxidative stress and map out what could be occurring in certain functional, certain regions around plaques and certain localized areas. So we followed the work from Backside's lab in a very nice PNAS paper where they used a genetically encoded oxidative sensor, ROGFP, It's being expressed in nerve cells here. And you can see with time, you can follow this in vivo. You can see that the plaque is stained in methoxy right here, it's blue. And you see it surrounding the tissue, you see these green neurons and neuroid processes that are intensely green. This is a measure of oxidative stress. The greener it is, it's increased oxidation. And this is the orange one that's more reduced. So there's an assay that was sufficient for monitoring how oxidative stress develops in a region around plaques. So we want to test the hypothesis: do microglial cells generate the ROS at a plaque that leads to this characteristic change in Alzheimer's that the postmortem studies has indicated is critical for the generation of dementia? Is this part of the second hit? So we have, it's not that complicated a hypothesis. First, we say that we should be able to see oxidative stress at the plaques using this another genetically encoded indicator, which I'll describe this to you. Base is similar to Rho-GFP. And monitor the GFP labeled microglial cells to see if we can see characteristic changes in these cells. And then we can delete the microglial cell using this receptor CSF receptor retainer antagonist PLX3397, which is developed by Kim Green, which will gradually put it in the feed for the mice, and the microglial cells die over a few weeks. And then we can deplete. So we have a way to deplete microglial cell and see if the oxidative stress is altered. There's a little nuance here in terms of looking at this type of experiment, is that the tool used, Rho-GFP, is an oxidation sensor, first identified by Roger Chen. This is a redox sensor. The problem, of course, is the microglial cells are also expressing GFP. They're not redox sensitive, but they do have GFP. So we use this sometimes, but we also use another new sensor that was developed by Matthew Tintama, which has a red output as well. But just to make sure you understand exactly what the experiment is here, we have Rho-GFP, or this other sensor I'll show you in the next slide, that detects changes in oxidative state within the cell. It's genetically encoded, it's in the cytoplasm. The emission of fluorescence is not affected by the redox state, but if the Rho-GFP is reduced, it absorbs most of the light when you excite its 2-photon laser at 900 nanometer. And if it's oxidized, most of the excitation is 800 nanometer. So you do a scan first at 900 excitation and 800, and you ratio the output and have a measure for oxidative degree of oxidative stress. And this is a nice example of what Stefan was seeing in the lab, is have a normalized value. You put on a cytoplasmic oxidizer, it becomes more oxidized, and then this is reversed with a reducing agent diethylthreotol. So it's very sensitive, and you can analyze the degree of oxidative stress in cells by doing a ratiometric analysis. And ratiometric has a great power. You sidestep all the changes in the optical properties of the tissue, and you come up with the actual value of oxidative stress within the cell. And so the nuance, we use this new Rho-GFP construct, which has a red M-apple tethered to the Rho-GFP. And so by FET transmission of energy, some of the fluorescence output activates M-apple. So everywhere there's a Rho-GFP redox sensor, there's a red output. And the microglial cells only have green output. So we can remove optically the pixels with the red sensor and look at the microglial cells in isolation, just the GFP. And we can also look at the ratio of the two excitation ratios and see the change in oxidative stress of the nerve cells with changing the either by putting on the oxidizer or by looking around the plaques. Okay, so using this row G RFP2, we show that just like the backside paper, we can follow these plaques for extended periods of time and see the development of oxidative stress. So these are these dystrophic neurites that form around the plaque. The plaque, we didn't actually stain in this example, but we can see it optically. There's a plaque in the black spot here. The oxidative stress is apparent in these green neurite processes that develop in the tissue around the plaque. And the microglial cells are now shown in white here to show where the microglial cells are aggregating and coming into the plaque and then surrounding the plaque. Okay, so now we have a situation where we can look at the tissue. We can look at the ratiometric values for oxidative stress and see how this changes. And one thing we notice, I don't fully understand this, but there's regions of intense oxidation within this plaque. These neural dystrophic neurites are called. They're swellings of the axons. They're highly oxidized. The ones that are most highly oxidized are surrounded and intensely attached to by the microglial cells, which is a consistent observation. Don't fully understand, but this is very much attracted to the highly oxidized cells. And it's an interesting question. Why is it sticking to the nerve cells? But it's certainly a consistent factor. So we then did the experiment. We have the ratio values for what the oxidative stress is in these plaques around in the dystrophic neurites. We can reduce the number of microglial cells by feeding the animals PLX for a few weeks. And we can measure this decrease. This is some measures of counts of the microglial cells after either staining or looking at this is IBA1 staining. The nice thing about this Ross sensor, we can also perfuse the animal, and if you do it in a certain way, you can preserve the oxidative state of the sensor. So we can feed the animals PLX. There's a dramatic reduction in the number of microglial cells. And this occurred both in the control as well as there's quite a reduction in the 5X FAD mouse. So now we have condition that the microglial cells are attached to the really highly oxidized cells. We can reduce the numbers around the plaques. So is it up or is it down? And so we, of course, our hypothesis was that the microglial cells are generating ROS. You get rid of the microglial cells, it should increase the ROS signal in the nerve cells. And so our hypothesis, it would decrease the signal in the nerve cell, of course. So the result is the opposite of what we had predicted, is that the reduced number of microglial cells is associated with increased oxidative stress in the nerve cells. So there's examples here, like this is the advantage of using a ratiometric analysis. You have some quantitative degrees of this distribution of oxidative stress in the cells around the plaques. Okay, so it's up. And this was quite surprising. Another way of looking at this is a distribution of areas around the initial plaque in the center here. You can see the highly increased oxidative stress in the PLX-fed brains where there's reduced microglial cells as compared to the non-PLX-fed. So conclusion, oxidative stress is increased in nerve cells when microglia numbers are drastically reduced. So why is this? We're still not positive what the answer is, but it does kind of confirm this hypothesis by Candello from Crescenzo's lab. is that the microglial coverage of the plaques is actually a protective mechanism. You can see in some of the stainings where you see the plaque, they're surrounded by microglial cells that are adhering to the plaque. And it's kind of argued that this is preventing the A beta diffusing away from the plaque source and affecting the surrounding tissue. So the microglial coverage here, we did Stefan did an analysis of what the coverage is. You see the IBA1 to stain the microglial cells. You can see the plaque in the center here is surrounded by microglial cells. And the LAMP is a enzyme that's in lysosomes, and this is very highly increased in these dystrophic neurites. And what we find is that the coverage is down in the PLX-fed animals. So it's kind of in an indirect way confirms the suggestion by Candello et al. That the microglial cells, when they surround and adhere to the plaques, this is actually protecting the surrounding tissue from the effects of diffusion of stuff from the plaque, maybe a certain amount of A beta. And the reviewers of the paper said, well, that's true, you should be able to just inject A beta, see increased oxidative stress, and it was indeed true. Take the A beta from animals that are injected already with the ROGFP, it's labeled the nerve cells, inject A beta versus scrambled, and you see lots of clustering of oxidative stress in these little green spots, and you don't see this in the scrambled injections. So microglial cells have a protective role in preventing the spread. And one thing Stefan did find with the analysis of what is correlated with the oxidative stress is the increase in the lysosomal markers. With LAMP1 increases, lysosomes get larger, they're quite densely stained with LAMP1. This kind of confirms the hypothesis that the source of the oxidative stress, at least one of the sources of oxidative stress, is lysosomal dysfunction from these dystrophic neurites at these areas surrounding the plaques. Okay, so future directions. We have the mouse. We're getting some data, a bit of a surprise. We hypothesized microglia are the key players. Microglia, I've convinced you, can produce ROS under some circumstances, but in the plaque, it doesn't look like they are the players, they are the protectors. And so we still want to explore this system to figure out why maybe a more human system. The mouse is interesting, get good data, but we don't want to cure the mouse, we want to cure the human. We need the human cells. And so we have this great collaboration with Håkan Nygaard, who's also in the center, and he's been making different forms of brain organoids, and Stefan's been using the ROGFP to look at how this can be used to look at oxidative stress, and then we can use this for looking at markers, what occurs in the cells, in the astrocytes, microglial cells, and nerve cells is correlated with the oxidative stress that we could interfere with in the culture system and use this to then potentially may have a rational therapy for developing agents to reduce damage in Alzheimer's. So Nygaard Lab is developing patient-derived iPSCs, and we have a few examples that we're growing. Stefan's growing stem cells and then developing these into organoids. He was first doing organoids from the iPSCs, and this was definitely a problem because there's typically an ischemic core. Okay, there's definitely an ischemic core within the organoids, so you can't be looking at a damage effect when the damage is occurring just from the organoid. So what he's doing now is developing a neurospheres where the nerve cells and astrocytes are growing naturally, and then we're looking at the microglial cells being introduced by growing the microglial cells separately. So this is just an example of you can generate A beta within the tissue, get oxidized clusters, analyzed with the Rho GFP, and this can be tracked. The neurospheres is definitely the model that we're settling on, because this is, you can do the manipulations and introduce microglial cells. You manipulate the microglial cells, put them into the system, and it's a much better way to look at this initial cellular environmental change that's triggered by these A beta conditions. And right now, we're mainly adding A beta in this tissue, and we're looking at Rho GFP being expressed in different cells, or glutamate or calcium. It's quite a powerful system. And the neurospheres develop. By day 60, you have a good cluster of astrocytes and nerve cells in a very complex environment. And then Stefan has a way of then growing microglial cells, and he can add microglial cells to these or not. And certainly if you add A beta, there are distinctive changes in oxidative stress that you see these little punctate spots. And the next slide shows orange is good. Orange is kind of nice, stable condition. You'll see that this cell then transforms, become oxidized. It swells up, starts to look like a dystrophic neurite, and then it's lyses. So I think we have a model system to go forward where we can look at the players. We can do manipulations to look at what are the important triggers, and the microglial cells can be grown in a separate kind of a bioreactor system and added to them, and then they invade the whole neurospheres. It's kind of remarkable, the strategy. And then we can also look at microglia and astrocytes separately from the neurosphere. And we end up with these spheres, three-dimensional clusters with the nerve cells look reasonable. Astrocytes are quite diverse, extend throughout the neurosphere. And microglial cells are added at an appropriate time to look at the effects. So I showed you why I started working on microglial cells. That was an astrocytes on just glia. That was a lucky guess. And I would recommend, when you have a crazy idea, if you can't destroy it by looking through the literature, it's most likely true. So the students, pursue those crazy ideas. That really can pay off, and this paid off. Hopefully I brought your attention. There's another form of immunosurveillance on a local scale where there's philopodia coming out of these growing larger processes that are searching around the processes at a much faster rate and a much greater area. That dementia is what we're trying to cure. And dementia is not always associated with A beta itself. There's a second hit. There's a second component that's being driven, and then postmortem tissue points to glial activation as a secondary component that seems to be required for the dementia. At least it's correlated with the dementia. That the microglia activation can produce ROS, which can lead to long-term depression, but I don't think this is involved with Alzheimer's disease. And finally, ROS imaging that at the plaques in this mouse model of AD shows that microglial cells can actually be protective and reduce ROS at the plaque, because A beta appears to be causing the diffusion of A beta into the surrounding tissue is really bad, and having the microglial cells shield the surrounding tissue from this diffusion could be a beneficial effect. And I think the neurospheres, this I think is what we're going to focus on now. This will give us the tools for looking at some of these mechanisms underlying oxidative stress and other disease processes. So my lab LP is a key player, Ellie York. Ellie's Jingfei Zhang is on here. Lots of great trees. I'd like to give all the credit to you for all this work. Thank you very much. Three minutes for questions.
Apologize, this seems a little naive. Obviously, oxidative stress appears to play an important role in the disease, and obviously in patients that actually do get dementia. Have, say, antioxidants, small molecule antioxidants, been used as possible treatments? And if so, have they been successful?
So, you know, a lot of people argue you should be taking antioxidants. I take antioxidants actually, just so I think it's still doing okay. So, but I don't think there's been any successful pharmacological trials using antioxidants yet. And I think maybe it's easier to argue if there's a disruption in the natural pathway, because there's a complex glutathione, vitamin C, ascorbic acid pathway that's important. But just applying it may be not sufficient.
Hi, beautiful talk, Brian. There's so much great data. I might not remember everything correctly, so correct me if I'm wrong. So did you say that the TRR4 knockout does not affect the LPD phenotype?
Correct.
So then how does LPS work, bypass TRR4 and activate CR3? Do you know how that works?
So it's binding to the CR3 receptor.
Okay.
So that's the evidence, the knockout of one critical component. So you don't have a functional receptor, then the LTD is lost. And this will surprise us. I thought this was a TOL4 receptor effect. No, wasn't.
I ask a quick question about these two modes of microglial process motility. You've described them largely as search strategies,
Right.
but I'm just curious what your opinion is about whether or not there's also either of these strategies are important for localized microglial release of factors that may be influencing synaptic function. When you were describing the LTD data in the context of LPS, there was sort of the cloud of
Right.
superoxide, but do you suspect that there's any capacity for movement to a particular region that's involved also in localized release of signals that can also impact neuron
That's a great
function?
question. I have absolutely no data whatsoever. I would entertain any hypothesis.
Any speculation based on
So
having studied the motility of these cells for so long?
are they releasing things as they're passing by? That's a great question. We have no data, but I saw a great chat today on exofurs, and you know, we do see strange things coming out of microglial cells in cell culture. It's very hard. I think it's very hard to see this in vivo or in a brain slice. But this kind of inspired me. We should be keeping an eye on maybe things being released. That's a great idea. But if something's being released not in a vesicle, like we wouldn't see it.
So thanks for that talk. It was great. It seemed like you thought that the activated microglia would be the source of ROS,
Right.
but then ended up concluding, in fact, they're protective. But I'm not sure what elicits the switch from them being the ROS generators to the protectors. Was it clear to me?
I can say I don't have an answer to that question. It's a key question. Now, this circumstance where they're ROS generator, there's a metabolic change as well as there is an enzyme activation. The NADPH oxidase has to form an active enzyme in the membrane and then releases ROS in extracellular space. There's also metabolic regulation. That's why the hypoxia and then also replicated by lactate shows that there's a metabolic link to how quickly that system is driven. I mean, the brain does have antioxidant capabilities. And I think that's it's a two, in this case, it's a combination of metabolic disruption as well as activation of the enzyme. That's what I would guess would be the critical two components. We don't have that in the Alzheimer's.
Okay, thanks. So in reference to that question, I guess there's varying degrees of evidence that some neuronal terminals can themselves express NOx, and certainly other cells around does A beta stimulate NOx in these? And have you just treated the whole thing with apocinin and see if that neuronal NOx goes away in the absence of microglia? I mean, neuronal oxidation goes away in the absence of microglia.
So we haven't actually seen lots of distribution of NOx in nerve cells. So we haven't seen much of an effect on these conditions with the apocinin. So I think we need a better assay system, because you start to look through literature. We have we generate a list for what could generate ROS. It's a long list. It's not only active enzyme activation, but it can also be maybe disruption of the astrocyte supply of glutathione or the antioxidant capabilities. So we're trying the list, running down the list. I think we need a good assay system. This is why I'm quite excited by the Neurosphere approach, that this will give us a tool to correlate enzyme effects, knockouts, et cetera, with changes in the oxidative stress in the tissue. So I don't have an answer to that. Great question, though.
Hi, Brian, that was great. Thanks so much. Yeah, okay. Yeah, thank you. So my understanding of the sort of microglial process extension, the major one, has always been that it was following an ATP gradient.
Right.
And that's why it had directionality. But your fine process movements, which are on a much smaller distance scale, they also seem to have directionality. But over that distance scale, you wouldn't expect an ATP concentration gradient. So why is it that they don't grow backwards?
They grow in all directions.
But all the arrows were forwards. Was that?
So that's the I couldn't get rid of that video.
Okay, so they grow in all directions.
They grow in all directions.
Okay, well, that makes me a lot happier.
I'm glad to make you happy.
Yeah.
How far back do I have to go? There it is. Okay. So here, there's no directionality. There's directionalities for the process, the large process. No directionality. We have
that bit was obscured.
That was obscured.
Yeah,
Yes,
okay,
that
good.
was terrible. I couldn't get rid of it. So you just see it's all going in all directions, backwards as well as around. So a minute later, another minute and a half, and you have this distribution scanning with a huge region. All areas of the brain have this. The Philopodia, the velocity is way, way faster, and the size is way, way smaller. The nanometer, 100 nanometer versus a few microns, and it's going maybe 10 times faster through rounded tissue. So we're just exciting this distribution, little sensors going around, probing around.
So that fits with the local surveillance model that you mentioned. Okay, good.
Yeah, so this is a local surveillance, yeah.
So there's a different molecular trigger.
Yeah, this is triggered by decrease in cyclic AMP. This is part of the signal. There's a decrease here, and this drive of P2Y12 is quite different. You have compartmentalized cyclic AMP. So we show that if we break down the compartmentalization by inhibiting the PKA and the phosphodiesterase, it's all over. You see this Philopodia everywhere. So this the localized cyclic AMP concentration is what's driving the Philopodia. And if we break that down, it's the whole cell is Philopodia rich all throughout the cell. So there's a next second player in terms of searching for targets.
Same front.